Hi everyone, in this video we're going to continue our discussion of protein synthesis. So last time we went over this flowchart and we started with the structure of DNA. Today we're going to go over DNA replication as well as the structure of RNA. So let's talk a little bit about how DNA fits inside of a cell. So human cells, you can imagine, are really, really small. DNA strands are really long. They can have, multiple nucleotides that stretch out really far. So we need a way to kind of package them into a smaller structure. So the way that DNA does this is through a chromosome. So a chromosome kind of looks like an X, and there's two pieces of a chromosome. So there's a pair of chromosomes basically that join together. So a human cell contains a total of 46 chromosomes or 23 pairs of chromosomes. And you can see those off to the right. So if you've heard of X and Y chromosomes, those are the chromosomes that determine your gender at birth. That's what chromosomes are, right? They kind of determine something about you, and that's because each chromosome contains one molecule of DNA and histones, which are small proteins that help DNA form this chromosome structure. So off to the right, you can see our double helix for DNA, which we talked about last time. Once that forms, the helix can actually kind of coil around, and it uses these histones to form this structure. And eventually, it wraps around again until we get this chromosome structure that's shown up above. And again, it kind of looks like an X. So the DNA within this chromosome contains segments called genes. And so this is the fundamental unit of heredity. And each gene will direct the synthesis of a specific protein. Now, human cells can contain about 25,000 genes, so you can create a lot of different proteins from DNA or these chromosomes. Now, let's talk a little bit about DNA replication. So an exact copy of DNA must be made in order to pass information on to new cells of a new generation. So there are a lot of areas of our body where we're constantly making new cells. For instance, we make new skin cells about every 2 to 4 weeks. We make new cells in our stomach and intestines pretty consistently. And there's many other examples as well. So if we're making these new cells, we need DNA to be able to kind of transfer itself to those new cells so we can make proteins or other, various information that our body needs to survive. So what happens in order to make these replica of DNA, we start with the parent DNA. And remember, there's 2 strands within that DNA. Now, if you imagine this like 2 ribbons that are kind of wound around each other, let's say at one point you start to pull the ribbons away from each other and you create a little opening. That's where replication will start to occur. So new nucleotides are going to come in and match up with the nucleotides on the parent strand. So maybe we've got A, C, T, G, or something on the parent strand. So the new strand would want to match that and complement it, right? So then we'd have C, A, G, T on the other side. So that happens for both parent strands where we formed that opening. Now you can do this at multiple points along a DNA molecule. So you could have replication occurring at multiple points. Now that continues until eventually we create a whole new copy of our DNA. So these are called daughter DNA, and each strand here, or each molecule of DNA, is an exact replica of the parent DNA, hopefully, if everything went right. All right. So the new DNA molecule that's produced, so the daughter DNA, has one strand from the parent and a new complementary strand. So here's more of a zoomed in picture of what's going on with DNA replication. So again, we have our original parent DNA. So it's almost like two ribbons that are kind of wound around each other. So we've got strand 1 and strand 2. And you can see that A is always paired with T, C is always paired with G, and vice versa. So now those parent strands are going to start to split apart so that we can put new nucleotides in. And those new nucleotides are in purple here. So complementary or matching base pairs will come in to form the complementary strand or the new strand until we have two daughter DNA molecules. So this is a really important process in our bodies. Now, how does this actually work? Like, what gets the whole process started? So there's an enzyme called helicase, and that is going to open up the strands of DNA. So we call that or we say that a replication fork has formed. So you can see that in our picture off to the right. Then in the next step, DNA segments, which are now known as Okazaki fragments after a scientist who did a lot of research on this. Those DNA segments are then synthesized by a DNA polymerase. So this is another enzyme that's helping to catalyze the process. So you can see the Okazaki fragments off to the right. And we're starting to form these new little segments of DNA. Eventually, those Okazaki fragments are going to join together by another enzyme called DNA ligase. And notice that all of these enzyme names end in ASE, so that's how we know they're enzymes. But eventually, once we form this, new strand here, we're just going to keep unwinding our DNA until we create two new copies of that DNA. So it's a very complex process, and it requires many different enzymes in order to occur. So sometimes it's amazing that we all function, right? There's so much that goes on in our bodies. Now, this process became very important as well in areas such as forensics. There was a scientist, Carrie Mullins, who developed something called PCR, or polymerase chain reaction, in the 80s. So PCR is a process that mimics the natural process of replication. So what happens is you can take DNA and basically put it into this PCR machine or instrument. You also add some DNA polymerase, so that enzyme that helps form new fragments. This also requires a cofactor, magnesium chloride. Remember, a cofactor is something that helps an enzyme function. So this helps the polymerase do its job. Then we have 4 nucleotide building blocks. So remember, we've got those 4 different bases that we need in order to build up DNA. And then that mixture goes through a 33-step replication cycles, and you can amplify or create multiple copies of DNA. So there's a theoretical amplification factor of 1 billion. So the general process is that you heat your mixture to 94 or 96 degrees Celsius for several minutes, and that's going to unravel your DNA into those single strands that we talked about. Then you cool the mixture at about 50 to 65 degrees Celsius for several minutes to allow primers to hydrogen bond to the separated strands. So we're kind of creating those Okazaki fragments in a way. Then you're going to heat again for several minutes, and DNA polymerase will synthesize those new strands. So you'll end up with multiple copies of the DNA. So again, this is really important in areas like forensics. So in forensics, for example, if you're trying to solve a crime and there's a little bit of DNA at the scene, you can amplify it or create several copies so that you can run more tests or put it into a database and figure out the full sequence of DNA, etc. So you can also use PCR to detect mutations associated with a genetic disease. You could also use it to detect the presence of unwanted DNA. So this could be bacterial or viral. PCR is really quick and simple to do. So what's also great is that you can use it on DNA samples that might have degraded. So again, forensics uses PCR quite a bit. You can also use PCR on extinct animal DNA, mummies, for instance, or even ancient insects that are trapped in amber. So you could try to sequence DNA using this method as well, and try to figure out, different information about various ancient mammals or insects, for example. Okay, so now that we've talked about DNA replication, let's go into the structure of RNA briefly. So remember, RNA stands for ribonucleic acid. And we said previously that ribonucleic acid, or RNA, differs from DNA because it uses ribose sugar instead of deoxyribose. It also uses the nucleotide base uracil instead of thymine. And then beyond that, RNA usually only has one strand that kind of wraps in on itself, similar to how a protein might form a beta sheet, for example. So DNA has 2 strands, but RNA often only has 1 strand. And you can see down below, we just have 1 strand here, and it's just kind of looping around until we have a section that forms hydrogen bonds with another section. So if you get perfect alignment here, you'll have G and C lined up, U and A lined up, etc. So that's one of the main differences for RNA from DNA. Now, there are 3 types of RNA. There's messenger RNA, which we abbreviate mRNA. So this carries genetic information from DNA in the nucleus to the site of protein synthesis in the cytoplasm. So within our cells, we have something called a nucleus. So that's kind of the center, I guess, of our cells. We won't go into too much detail on cells here, but The cytoplasm is basically the area within a cell that's not in the nucleus. So messenger RNA just kind of delivers information from one place to another. That's the main point. Messenger RNA does have a short lifetime, which is less than an hour. So it's synthesized as needed, and then it rapidly degrades into its individual nucleotides. There's also ribosomal RNA, which is abbreviated rRNA, and this is the main component of something called a ribosome. And ribosomes are important because they're the site of protein synthesis, and we'll talk more about ribosomes later. But rRNA accounts for 80 to 85% of the total RNA that's in a cell, and it accounts for 65% of the ribosome structure, and the remaining 35% is protein. So again, we'll talk about ribosomes later, but again, the ribosomes are where protein synthesis occurs. And then finally, there's transfer RNA, or tRNA. So this actually delivers individual amino acids to the site of protein synthesis. Now remember, amino acids are the building blocks of a protein. So tRNA is what takes an amino acid and delivers it to the right spot to make a protein. Now, tRNA is specific to one type of amino acid, so cells will contain at least one specific type of tRNA for each of the 20 common amino acids. So each amino acid has its own specific transfer RNA that can deliver it to where a protein's being synthesized. Now, tRNA is the smallest of the nucleic acids, so it only has 73 to 93 nucleotides per chain. And this is relatively small compared to, for instance, some DNA. tRNA also has two regions that have very important functions. So there's a region called the anticodon, and this helps tRNA bind to messenger RNA during protein synthesis. So let's just look at the structure of tRNA real quick. So you can kind of see where tRNA kind of looks like a T, so that's kind of convenient. But it sort of kind of wraps around on itself. So it has some of these loops, but then areas where it does complement itself. And it creates, this kind of T structure. And there's, again, that anticodon site, CGC. So cytosine, guanine, cytosine. And this is like a little code that tRNA uses to say, oh, I can attach to messenger RNA using this site. There's also another site, the 3' end of the molecule, which binds to an amino acid. So at the top here, on one end of tRNA, the 3' end, that's where an amino acid can attach itself so that tRNA can then transfer it to where a protein's being synthesized. And remember, there are specific transfer RNA for each amino acid. So each of the 20 amino acids has its own tRNA. All right, so we've got a similar structure here to our nucleotide, right? So remember, RNA has ribose, it's sugar. It has a nucleotide base, and then phosphate. So this is our kind of tRNA structure. And then on the 3' end, so the third carbon in the ribose structure there, we have our amino acid. So remember, in an amino acid, we have the carbonyl group, or the carboxylic acid group, I should say. We've got the amino group. We've got our alpha carbon attached to hydrogen, and then we finally have the R group, the side chain. So we can also abbreviate this structure as shown on the right. So here's our amino acid, and then we can just say, here's tRNA, and we'll just draw that little T structure that we saw. Okay, so again, tRNA, its main purpose is just to transfer an amino acid to the appropriate place in a protein. All right, so let's just review what we've learned so far. So how does RNA differ from DNA? And then what are the 3 types of RNA? So see if you can remember these on your own. So you can pause the video, think it over, and when you're done, we'll go over it together. Okay, so let's start with the first question. How does RNA differ from DNA? Well, there are multiple ways that they differ. The first way that they differ is we have ribose instead of deoxyribose in the nucleotides. Additionally, we use the nucleotide base uracil instead of thymine. Let's see, RNA is usually singly stranded, whereas DNA has 2 strands. So that means that RNA can kind of double back on itself. So it might look something like this, where you've got those hydrogen bonds between the same strand of RNA, instead of DNA, where you have 2 strands that are hydrogen bonded together. So what are the 3 types of RNA? So we said that we have messenger RNA, mRNA, where m is lowercase. So this is going to carry genetic information from DNA to the site of protein synthesis. So it literally is a messenger, right? So it's carrying information from DNA to the site of protein synthesis. Then we also had ribosomal RNA, or lowercase r RNA, and this is the main component of what are called ribosomes, and that's where the site of protein synthesis exists. So that it's the main component of ribosomes, the site of protein synthesis. And then let's see if I have enough room for the last one, which is transfer RNA. So transfer RNA, or tRNA, delivers amino acids to the site of protein synthesis. Acids to the site of protein synthesis. So again, there are different types of RNA, but they all play a very important role in the synthesis of proteins. And remember, RNA is kind of the go-between for DNA and proteins.